I think you can see the screen now. Um, but I want to call this kind of like not a lecture, but more like a workshop about how to use Benchling. I call it Benchling Basics, and also like a little bit of cloning, which I really want to use the word like cloning by scenarios because like we can like discuss about like what is the scenario that you end up with when you're doing like the DNA cloning or something like that. So just a disclaimer, this video will be this presentation will be recorded. Um, and I'm planning to share it on YouTube so that like others apart from just our group can access and like take a look at it as well. The materials will be turned into a PDF so that like, people can access to these like public Benchling files from the links that was put there as well. Other than that, I think you can expect to learn how to do some like basic operation in Benchling, and also like take it as a chance to discuss about like what is the way to do this like operation, or like what is the necessity, or why do we have to do it in these kind of scenario or something like that. All right, let's start. So the idea behind this workshop is because like when anyone is coming to molecular biology or like is coming to like synthetic biology in general, you have to work on the molecular biology tools that are similar to the text editor. So when you're taking notes, or like writing a document, you may be using like a Google Doc or a Microsoft Word for the text editor. But in molecular biology, they're a little bit different because like most of the information that was stored are not in like the Roman alphabet, are not like A to Z, but they are rather like A, T, G, Z. They have like two-stranded. So like the tools that were used for these exploration will be a little bit different. And our labs have been using Benchling for some simple molecular biology operations. And one thing, the one reason that we've been using it is because Benchling was made free for academic users. If you're, turning into an industry researcher, you may, like your company may have to pay for it. But so far, it has been a pretty good tools for us in the past few years. I mean, for me, for at least like 6 years so far. And one thing I want to like, mention before we get too far into our presentation is that like, I'm very pro doing in silico cloning or like in silico operation in any tools before you do the actual experiment, because sometimes it saves a lot of time. When you know that the experiment is going to fail before you actually do it, that's going to help a lot. But sometimes you don't really know how to do it properly, or like, how to use it to the base of this operation. So like maybe like, the process today can show you a little bit on like what is possible out there for using these tools. So the outline would be like we would do, I would present about like a little bit of like intro on why do we need these like specific molecular biology tools. On the Benchling side, we will show like, how to attach primers, how to do PCR, and then we will navigate some simple assembly research in Benchling. I think I plan for like, restriction digest, Gibson or Infusion assembly that we have been using so far, and also for the Golden Gate. And along the way, we discuss some scenarios when we do cloning, so that like we know if we do this kind of cloning, if it works, it should give you whatever results that you be clearly, it will be clear to you when you're doing the cloning, something like that. Like, and one example would be like you're cloning something that you're putting the green fluorescent protein in, then you should see the green fluorescent protein. If you see the white one, then that would mean something different, and we can talk about that. All right. But the most basic thing, I mean, just in case someone just come and like watch this recording, most of the biological information were stored in the form of DNA. And like when you take a look at this DNA here in biotech or in SynBio, we usually use the plasmid DNA as our order operation. Most of the things that were changed in this plasmid system is actually the gene of interest. Just in case there's any question about these processes, we had a previous lecture, kind of mini lecture about plasmids. So, that would be a good place to take a look at if you're you wonder what is the plasmid or like what kind of plasmid you should be using in your processes. But with that, if you have a plasmid, we can consider DNA as a Lego piece as well. So this is a very beautiful figure published in ACS in Bio in 2015. It's one of the first paper that I learned the modular cloning in grad school. So this paper is about molecular cloning yeast toolkit, or someone may have heard about it as like Moclo YTK or something like that. We can consider these DNA pieces as a Lego, so that like they can connect with each other using the specific end that we joined together. The figure that you've seen right here is like, at least 8 parts that were combined together, where these parts will have different functions. So just to not get into like too much of details, these parts were made by like, having a lot of pre-made system that was available here. Then you combine them into one particular setup. You can even plan to combine different parts together at the end, where you can do more than just one assembly, but do like multiple assembly to make like a plasmid that might be bigger or having more function. So like on the right hand side, you see like the cassette plasmid and the multi-gene plasmid. The multi-gene plasmid is pretty useful, where you're trying to put something into the same plasmid with the same like set of like materials that you have, but like can do several things at the same time. So again, I would encourage you to think about DNA as a Lego pieces, so that like you can combine them together. One concern that I want to share right now is that like in synthetic biology and in molecular biology, there is a lot of context-dependent behavior that's going on. So when you put things together like this, where if you put it in the order of like 1, 2, 3, but change it into 1, 3, 2, there might be some differences. So that would be something to be concerned. But we skip that for now because we want to keep it very simple. And another background is that like we talk about DNA, right? We probably want to know how DNA was made. So there are actually 3 simple processes that scientists have been using to make DNA. The first one is just like using biology to make the DNA. You can use like DNA replication process, put some DNA into the bacteria, let them grow, divide, so that you have a lot of DNA accumulated in the bacterial cells, then isolate them out. That was pretty cheap, but it's not that easy to modify the DNA that were in the bacteria. So the cloning, molecular cloning, is one of the way that we use to modify the DNA before we do these like replication. The other thing that we have been using is to use kind of enzymatic or a chemical reaction. And polymerase chain reaction is the name of it, where you can do some simple modification to your DNA. This is relatively cheap. Every biotech lab has been using PCR. And if you don't have access to PCR, that will be very difficult for you to do any like synthetic biology or biotechnology work. And the third one is something that has been like, a pretty big disruption or disruptive movement to the field, because like it's getting a lot more cheaper, even though it's a relatively high cost process, which is like trying to synthesize the DNA through the chemical synthesis. The good thing about this is that like you can just like order some piece of DNA, and we just come in the front of your lab like in not too long, and you can ask for almost anything as long as it passed through the synthesis and the safety processes that they have. So instead of doing PCR to get a new piece of DNA, you can just order it, and there will be some situation where ordering the DNA is actually cheaper than PCR. It depends on the time that will be consumed, or like the other kind of processes that will be involved as well. The chemical synthesis process involves like the synthesis of the oligonucleotide or the short pieces of DNA that it has a very fast producing process. Manufacturing process as well. So, if you want to know more about this system, you can take a look at, some oligonucleotide synthesis out there. But with that, when we have the DNA, we'll probably like do some operation on this DNA. So these are some lists of cloning or kind of like manipulation of DNA that we can use. I'm going to go very quickly about it because we will do the actual in silico work on them again. But one type of cloning is by restriction and ligation. It's kind of like old school. You just like take the Lego pieces, cut them so that they have these kind of hinge together, and then they can connect back specifically to whatever hinge or like connector that they've got here. So this one would be based on the sticky ends. That's the specific word for us when we use for these kind of like restriction ligation processes. Some of the restriction digest will generate like a blunt end, and that will give you a different kind of like design strategy for doing these molecular cloning processes. The other way that people have been trying to standardize, and this is like one of the movement that makes cell biology be more systematic these days, is by using some other restriction cloning still, but like make it more an assembly strategy. So this is the BioBrick assembly strategy, where you have like these common cut sites on the left and right hand side. The good thing about this is like you can connect part A and part B and have them be in frame. So like you see that these scar were 6 bases, and they're actually in the same, they're actually like referred to just 2 codons. And that means you can actually like make part A, which is maybe 1 protein, and part B, which is another protein, to make a fusion protein right away with this assembly strategy. So, this assembly will regenerate these cut sites as well. And that was one of the methods that synthetic biologists have been using in the past 10 years. These days, we probably don't really need these complicated design anymore, because like synthesis and like PCR is a lot more easier. All right. These are probably like the process that we've been using a lot, which I would like to call it overlapping sequence assembly. It's not, and I didn't make it up, but like when we call this assembly, is based on the overlapping sequence. And most of the time we just call them Gibson assembly, because that's this was made, it was designed originally by Gibson at JCVI, but there are other methods that use overlapping sequence for these assembly as well. And the good side of it is that like it's usually scarless, because you can design any sequence to make an overlapping sequence like this. And it gives you a very high accuracy. So most of the time, if you get any positive results when you are putting these DNA into the bacteria, they are likely correct. This is another process that our lab at UW has been using a lot, called infusion cloning, and there are some differences here. The key differences are that they use only 1 enzyme rather than 3 enzymes that were used in the Gibson. And by using just 1 enzyme, it sort of like prevent some undesired processes. For example, like if there are some ligase that can connect the piece of DNA in the Gibson assembly together, you may have like different scenarios that come out of the Gibson assembly. But since there is no ligation in the infusion process, the background, the false positive, or like the other scenario that can occur will be avoided. So if you want to know more about these processes, you can take a look further. And this is the pamphlet that came from this website, Chair Biology. They have a very very high quality infographic right here. I'm not going to go too much into it, but I would encourage you to take a look. One thing to keep in mind is that like most of the reagents that were made available commercially for these processes are not that cheap, but it's also not that expensive. So you may want to check if you were to do just like like simple ligation or these overlapping sequence assembly. It depends on like how fast or like how cost efficient you want to be with this kind of operation. There are also some PCR-based modification of the DNA. Like the easiest example would be like to do mutagenesis by designing the primers, do some substitution, deletions, insertion, or even large insertions sometime. So I just want to put it up here that these strategies are also available. But it has some limited engineering property, because it's usually engineering only at one site at a time. And there are also like the Golden Gate assembly, but like I'm not going through that right now because like I just mentioned it earlier that they're actually like an improved process of the restriction ligation. And again, I'm going to revisit a little bit of the plasmid architecture here, that whenever you are trying to clone something, you probably need three pieces of DNA to make the plasmid that you put into any bacteria. Like you want the replication part of the plasmid, you want the selection part of the plasmid, and also whatever genome interest you want to put into these like new plasmid DNA. But most of the time you'll be focusing on changing this gene of interest because you want to introduce new thing to your organism of interest to see what is the difference or like the new property that were generated by introducing these new genes. All right. The thing about plasmid is that like putting plasmid into bacteria is a relatively simple process. It could be done in like, several different ways. But what we have been doing so far is usually the transformation process that can involve, either the chemical transformation or the electroporation process. For chemical transformation, we usually like put some chemicals into the bacterial solution, then make the change of temperature to instigate the DNA taken into the bacteria, something like this. Or we can use electricity to like shock the bacteria so that they are stressed and like they make some pores in their membrane and then let the DNA get in. So these are two simple processes that can be used for the putting DNA into the bacteria. I mean, yeast can use like some similar processes as well. Most of the time, you will not care too much about the transduction or transfection if you were working in bacteria, because like those were more likely the process on the mammalian side. But yeah, you heard a lot of transformation or conjugation if you work in the prokaryote biology. All right, let's get to the real thing. Now that we are trying to make the DNA sequence, I want to show you first that like whenever we make new DNA, we also need a way to validate whether the DNA that we have are correct or not. And you can actually do that by some of the sequencing technology. I'm not going to come back to the sequencing anymore, but there are actually like three simple processes so far that were used in the sequencing technologies. The first one is maybe like the very first DNA sequencing in history, which is like Sanger-based sequencing, and they will give you these kind of results where you'd have different color that signify what is the base or what is the alphabet of the DNA in the order how you're gonna read it. The next gen sequencing is like a process that were designed to read the DNA in a massively parallel way. Imagine like you take the chunk of DNA, split them into smaller pieces and just read them, and then use the computational process to stitch them together afterward. While the third one, which sometimes people call them the third generation sequencing, is a long read process where you don't read just like a small chunk of DNA, but read a long sequence of DNA and then again combine them later. So both the next gen and third gen are like capable of reading something that was like full plasmid or even the genome level of DNA. So if you're interested in that, please feel free to take dig further into the system. But since I show you this kind of snapshot, I think you probably wonder like what kind of software or like what kind of system you should be using to read this DNA information. And one simple take I would like to show here is that like if you see DNA, the DNA data that were stored like online, you probably see them in various formats. The very simple format would be like the FASTA, where you just have the DNA sequence, only just one strand most of the time. And with that, you can like convert it into something else by like doing some downstream analysis on that. The second type would be like something that was like GenBank or like GFF files that have not just the DNA information, but also have the annotation information. like what is this part of the gene or this part of the DNA referring to which gene or which protein or which regulatory element that the organism has, or maybe like there might be some kind of format that were relatively proprietary to the program, like this .dna that I believe was the file system from Snapgene. And you can probably take these kind of formats and like use it further in a lot of softwares, because like it's similar to like when you're using Windows and Mac, people are trying to make them compatible between different software. So most of the time, if you get the information that were stored in whatever kind of DNA information, it will be convertible to a certain extent. So what do they usually look like? If you take the FASTA file of E. coli genome, it will look like this in the notepad. You just see a bunch of text in ATGC information. If you try a little bit harder and take the GenBank file and then force Microsoft Word to open it, you see this kind of information where they have like, oh, this is a name. There are some features of it. These are the position and also the sequences of each part. And at some point, if you have a good software, you can actually take these information and turn them into something that was easier to visualize, something like that. So what I meant by that is like by using this kind of proprietary software like Snapgene. This is the program called Snapgene Viewer. If you were to use the full function of Snapgene, you may have to pay for their subscription fee. But we can use this Snapgene Viewer to just view it without paying, because they have a free trial available for you to just use it. But of course, you're not going to get a full function of it. The other ones is Benchling, which is a cloud platform. I'm sorry, I forgot to mention that Snapgene is actually like installed on your computer. So you can just work on it anytime. But if you want to use Benchling, you may need internet to edit anything on the cloud. So this is like what you see when you take a look at Benchling. As you can see right here, they look not too different, but the DNA will be visualized as double-stranded DNA. And you also have some annotation related to like the DNA you're looking at. And as you can see, it's cyclic, it's circular, because this is the E. coli genome. If you just read at these like Notepad or Microsoft Word, you probably don't know whether it's circular or not. But there will be some annotations that were encrypted in here that shows whether like the DNA is circular or not. All right. So again, these are the link to access the software. I'm not gonna focus too much on Snapgene Viewer. There are probably some information out there that can help you navigate if you prefer these tools, or there are some other tools like Ape, a plasmid editor, Genius, or other tools out there. But again, like we've been using Benchling a lot, and it's free for academic users. So we're gonna focus on this one. And after this, we're gonna do some demo. All right. Let's think about this kind of plasmid where you have three pieces. You have like, the CoE1 or as the replication machinery right here. You have MR, which is stand for ampicillin resistance marker, where you can use it for the selection in the bacteria, for example. And you have this part called GFP, which is going to translate into the green fluorescent protein, and it's going to make the bacteria green, for example. If you want to make this kind of plasmid, you can make these by using some other existing parts, do some restriction digest, do some PCR, and try to combine them together. But what if we don't want to do any PCR? Can we just order everything through the chemical synthesis? The answer is yes. So let's make an assumption by, let's order these three different parts as a double-stranded DNA, so that we can just assume that we do PCR for all these, and then try to do some operation downstream to connect these things together. All right. Here, what I'm going to jump into Benchling next is that I'm trying to take these three DNA pieces, cut them with the appropriate DNA cut site that was shown right here, and then just try to connect them together into the plasmid using these three synthesized DNA parts. All right, we're gonna jump to the Benchling. All right, I hope you guys can see my browser now. So I have this benching project here, and one additional thing about Benchling that I wanna mention is that you can actually make something like the text editor here as well. Ooh, you can see the Thai language here because I install this one using Thai, but let's see. So you can actually take some notes here as well. First, we're gonna try to do ligation of 3 pieces of DNA. And I actually pre-made these DNA already. So they're actually something that resemble the different part that I show you in the PowerPoint. So let's get rid of all other tabs so that we can focus on this one. Close all the tabs. So along the way, you see some function in Benchling that I'm showing as well. I may not talk about it, but hopefully you can see how the clicking is going, and that may help with the process. So we have these 3 DNA piece in Benchling format already. We have the first part, which is the GFP, the second part, which is the MR, and the third part, which is CoE1 or the replication machinery. What we want to do is to combine these 3 together. You can sort of copy paste this thing. Like you can see if I hit Control C, you can copy this to clipboard. You can paste it right here, and they can take the feature and copy it right away here as well, right here as well. But this process is less straightforward. In the how do you say it? They don't provide a lot of evaluation process when you do this kind of copy paste, because you can copy and paste or do the edit to any DNA as much as you want, right? What I'm gonna show you is that I'm gonna use the assembly wizard right here. So if you click on the bottom right there on the assembly wizard, you will see that there are 3 assembly tools available. The simplest one is digest and ligate. So I'm gonna do this digest ligate real quick by connecting these 3 pieces together. When I start with that, it actually showed me these choices of what should be the backbone, what should be the insert. Technically, it doesn't matter too much if you have all of these be an insert. But let's do this by use the original replication as a backbone, because that's likely one of the most important thing when you're trying to clone something. If they cannot replicate, then your plasmid usually cannot stay inside the cell. So for this one, we're taking a look at the backbone right here, and we're selecting the fragment, and I will click at this restriction enzyme box 1 and this enzyme, and maybe set as the backbone. I hit set from selection here. Then it's giving me this kind of instruction that, hey, it seems like this is your sticky end, or your end of DNA that was shown right here. This SGS1 give this kind of sticky end. This box 1 give you this kind of blunt end. I have 2 insert, so I'm gonna add 1 more insert right here. The first insert I'm gonna add is this 1A. Okay, we're gonna cut by SGS1 and also KFL2 right here. Sorry, KFL1. Similarly, I just hit set from selection. And it actually showed you that the SGS1 part right here, from this piece and this piece actually meshed together. So it means that this is compatible. And if you do it by hands, you have to ensure that your copy paste is matching properly. But by using this wizard that they provided, you can actually match them accordingly by letting the computer help you validate this process as well. Again, let's go to this last part. It actually highlights the piece that were used in the digestion here as well. So we have to use this KFL1 as one of the cut site. If you just randomly click and go to this, FAC1 and hit it, it's gonna tell you error. You can't stick these 2 things together. because they don't have the compatible sticky end. You have to choose a different one. So let's reset, abort it, and I'm going to choose the proper one, which is box one instead, and set from here. Then it's actually telling you that like there's no error. Looks like everything checks out. And if I hit done right here, I'm going to assemble them into CoU1, MR, SFGP. And it's going to generate you a new file that has these different pieces together. In the assembly history, it's going to tell you that you have these 3 pieces together. Maybe you have another record on your notebook that like this 1C, 1A, 1B were synthesized by whatever method. And then it will combine with these 3 fragments together using the restoration ligation. So that's one example of how to use Benchling to do the cloning using the ligation. This looks like, you know, something very simple where you synthesize the DNA. But what if you try to do something that was like a little bit different? Again, just for the context, for the scenario here, if you have the bacteria that doesn't have anything, they probably look kind of white like this. If they have this plasmid where you put them by transformation process and they have like the GFP, they should look like this green colony. And when you grow into the liquid culture, they should look like this. If you have GFP, it should be kind of green, like yellowish green. If they have nothing, they will look like this. All right. What if we want to convert one protein to the other? Let's say we want to convert the GFP part right here to another protein, another enzyme of interest. Let's choose RFP because the color is so different. And, you know, a lot of you who are working in synthetic biology probably like have heard about like, you know, some other fluorescent protein as well. And one simple process that we have been doing all the time in biotech is that like we take one existing plasmid and just change one part to the other. So what I'm going to tell you, what I'm going to show you right here is how to use Benchling to do the PCR by taking this piece of DNA from somewhere else, like from the other plasmid that has this MRFP, and do the synthesis or do the PCR to get the new piece of DNA and do the assembly. The assembly that we're trying to do here, we would do the Gibson style assembly. So let's jump to the Benchling one. All right. Here, I make it in the second folder. So we start with having, you know, 2 parts available. Let's take a look at this one. This is a plasmid that we made previously, right? By having the selection marker, the backbone replication part of the backbone, and also the SFGP. If we want to change just the GFP into the RFP, we can simply just like digest the SFGP part out and replace that with the MRFP part. So on the left-hand side here, I actually have another plasmid that has MRFP part right here. What I'm trying to do is that I want to make the plasmid that look like this by having almost the same thing like the first one, but change the SFGP to this MRFP construct. Maybe like a good example to show you right here is that like if you take these 2 things together and try to like compare the sequence between each other, actually this is another sequencing results. So let's try to compare this desired plasmid, desired sequence to the other existing one. So if we try to compare with the one that has just SFGP, and just go through the default here, you actually see that these new plasmid can be made by just changing one part right here. Like you can start changing from around here where the things are different, and then start changing again around here where things are different. So the actual way to do it, you know, could be done through different processes. The way that we want to do here is by doing the Gibson assembly. So I'm going to hit the Gibson assembly again. Let's do Gibson assembly. All right, let's set the backbone fragment. the backbone fragment of these, desired construct right here, CoE1 with MR selection that has MRFP part, we probably start with everything that doesn't have the MRFP. So you see, if you recall the cut site that we used previously using these SGS1 or whatever part that were available, we can actually select everything but this part. So in this case, I'm gonna select everything but the BioBrick part, so that it behave as the backbone for us. So I'm using some trick by like selecting everything that I wanna take. You can also do the other way around by just like click the thing that you wanna replace and do inverse selection like this, or you can do that again and you get like the backbone like this. All right, so the backbone that we are making is actually set by selecting the SPA1 and XBA1 cutting, and I'm gonna set this fragment like this. The insert though, we want them to be this kind of piece, right? We want them to look very similar to this kind of piece that we want to put in. But the real scenario is that we don't have this plasmid yet. We actually have to take the MRP sequence from somewhere. So the reason that I have BioBrick in the final part, the BioBrick prefix and suffix, like this kind of like part right there, is because we have this piece around. So I'm actually gonna start choosing from this BioBrick prefix to the BioBrick suffix right here, and then set this as a second fragment. So the good thing about this assembly wizard is that they will actually give you not just the method of construction, but also the primers that you should be ordered to make this Gibson assembly as well. So this is going to be CoE1 MR MRP. If I hit assemble, they'll ask you where do you want the sequence, the primer folder to be. The primer folder and everything. I'm just gonna put everything into this folder, and I'm gonna create it. With this, the assembly parameter is that you should be doing the PCR using these two primers, and you'll be assembling. You'll be assemble them with this method. So this yellow part right here were achieved by just digestion using XBA1 and XBA1 and SPA1 as shown, and then you have to generate this new fragment using the PCR, in which you get this green fragment that has MRP, and finally you can assemble them into this kind of fragment. Right here, it actually gives you two primers that you should be using, which is this primer 2 prime, PSE 101, whatever, that bind at the template, and also this one, which is a forward one that will combine. So what you can see right here is that like it seems like this assembly tool actually give you a duplicate piece of the insert. That was likely because when I choose the piece that I want to do the Gibson assembly, I include the prefix to be added as well. So it's sort of like double that into our final design here. So this pair primer will actually give you the double like DNA sequences of this BioBrick part. So this is something to keep in mind, and this is some kind of error that you may have when you're doing this kind of design as well. All right, but one thing I want to show you here is that if you click this primer and hit open primer in this sequence, this is the DNA oligonucleotide that you can order, and if you order through like the company that do a regular shipping, you might be able to get it from just overnight or just in a few days, depending on where you are. But this sequence that I just copy around, you can actually search for it, and you see that this sequence can be found in the desired DNA that you have. You can even do this function by create primer like this, and if it match any existing primer in the benching directory that you have access to, it will ask you whether you want to use whether you're going to use the primer that's already existed and just bind to it here. So you can actually, reuse the primer that you have ordered along the process, so that you don't have to wait for, let's say you want to do some new cloning, you can actually find existing primers and just use it right away. The other way to find the primers for the PCR is by use these primer attaching tool. So if we go to the primer on the right hand side, this primer button right here, and hit attaching, they will ask you you want to search for existing primers. You can actually search into all the projects right here. It's gonna take a long time, but let's just try it once. But I'm gonna also use this use selection so that it search for only this small part of the DNA that was available right there. And let's just try, find binding site spread. By using this function, if there are any, DNA sequences around in your, accessible notebook, you see what kind of primer that can be bound to these processes. So as you can see here, it seems like my lab has a lot of primers that can be attached to this general part. And when you attach it, sometimes they just show you some incomplete attachment as well. And that depends on the selection criteria that were shown right here. So again, if your lab or your team has a lot of primers in your catalog, you can actually use this search function. And I think this is one of the strength of the cloud, because you can actually find something that your friends or your teammates have, so that you can borrow those parts and just use it right away without reordering. So you're not wasting your money and also your time into using some of these common parts, for example. All right. So that's it for the assembly using the Gibson base or the overlapping sequence base. I'm going to go back to the presentation and get into the other scenario again. So after you have that, you actually, purify your DNA, right? You have this kind of this is one example of my gel where I do digestion. I have this kind of, band showing up. This is probably the closest to what we have, where we have, the big band, also the smaller band where you take the GFP out. And let's assume that this is the MRFP band where you do the PCR. You may have some smaller bands that came from some other non-specific PCR reaction, but you know that your desired PCR will have this kind of size around, let's say this is probably 1 kb, which is pretty close to the 1.1 base pair, 1.1 kb right here. You can isolate this DNA fragment and then combine them, and then do some chemical transformation, and you'll likely change your bacteria that initially turns green because they have GFP into something that was red like this. So again, if the scenario like this is not going according to your plan, let's say you get the white colony or the green colony instead of the red colony, then that means you actually just have some contamination of your original plasmid that have GFP still, or you just don't have anything and they just the backbone fragment may just fuse together without giving you any new insert. So this is a pretty good scenario in terms of the analysis of whether your cloning were successful or not. Most of the time you will not be lucky like this, because your cloning may not give you any visible color. But for some system that you guys are using, if they have color, you will be in a very fast-paced evaluation process like this. All right, so let's get into some real kind of what we've been doing in our lab. This is as a rough diagram that when we're doing any CRISPR experiment in our lab, we will have the part called the guide RNA that we're available as well. So there'll be at least the promoter, the spacer, the hairpin part of the gRNA, and also the terminator part that we need to copy over and over when we're cloning these kind of new gRNAs. So what I'm gonna show you right here is that if we were to clone a new gRNA, what is the process that we usually use to clone this new gRNA? So we actually use the Gibson base, but can we just design a new primers that can just get in right here and target some new thing? We can actually do that by just this simple, 60 base oligonucleotide order by ordering these primers to bind right here at part number 3. Include this part number 2 in the primer so that the primer will just change everything, and they will have some overlapping sequence with part number 1 so that we keep everything the same, but this part number 2 that was around 20 nucleotide. And this is very doable and it's very fast. So for example, if we have some reported genes and we want to do some gRNA that can manipulate these green fluorescent protein or red fluorescent protein. In my lab, we do the CRISPR gene regulation process called CRISPR-A and CRISPR-I. A stands for activation and I stand for interference. And an easy example of interference is that if you start with something that was green, so like, this reported gene, this plasmid, actually have 2 reporters. They have like medium expression of GFP, the green fluorescent protein, and the weak expression of MRP, which is like the red color. So if you just take it and just put into bacteria, they will have green color and not showing any red color because it's so weak. If you do repression and by getting rid of the gene expression of this SFGFP, or we call it CRISPR interference, you can actually turn this this system from green to white. If you do CRISPR activation on top of that by repressing the green color and also to activate the red color expression, you can actually turn the bacteria from green to white and then to red because they are accumulating this red fluorescent protein again. So this is an easy example. But what if we were to do the real kind of experiment? You don't really want to work with the fluorescent protein anymore. You want to get rid of these maybe like AMPC gene, which is stand for like ampicillin resistant gene in the bacterial genome in E. coli, for example. Can you do that with CRISPR? The answer is yes. So let's go to Benchling real quick and see if we can do it with the simple design we have. All right. So we're gonna start with this, 411, PCK 411, which is the plasmid that was available on AdGene. This gRNA right here is actually the gRNA that was used for the repression of the MRFP. But as you can see right here, they actually have this SPE1 cut site right next to the promoter. So that was where we designed them so that you can cut right here for any change. And there was like the BSP21 enzyme cut site right here that technically if we cut these 2 pieces, we can actually replace this whole gRNA cassette and change this 20 base part into the new thing. But before that, we may want to see like, what is the target that we want to get rid of. Let's say the MC that I just mentioned. If you get into this E. coli genome and search for MC, it actually show up right here. And when you do the design for the CRISPR knockout or CRISPR interference or whatever, the CRISPR system actually require the PAM site, right? So if you search for NGG, you can just like highlight this part and hit Control F, similar to how you use Microsoft Word, and hit NGG. There'll be some NGG sequence that show up. And this is the piece of DNA that can work as your PAM or protospacer adjacent motif. You need the spacer sequence to be right next to this PAM. And this is the spacer that I have designed previously to be around 20 base. And you can actually copy that and use it as the new 20 base sequence that you can design the new DNA in your oligonucleotide so that you can make the PCR and just do assembly again. All right. One important thing is that you need this DNA that is on the other strand. As you can see here, this direction of the gene that was read right here is on from right to left because the gene is on the other strand. So this is one of the important process that you have to be very careful when you're doing this design process. All right. This is a primer that has been used before. So I'm just going to do a quick trick by create the primer right here. I just copy the 20 base previously, and I'm going to change this 20 base into this new sequence. Seems like there's no primer like that before. I'm going to call it MC.RNA. Save it in the gRNA part. Now we have the new primer. With this new primer, though, you can actually pair this primer with another primer right here. Within the primer tab, if you click this one, you actually have to hold Shift and hit this again, and then you'll be selecting these whole DNA pieces, and then you can go to the pair system where you can link this primer, and then you can actually create the PCR product right here. All right. This is a product that I have. This piece is not RR1 anymore. Again, you see that like this previous RR1 was the template, but by doing the PCR, by using this primer, you actually change these 20 bases to something new, which is this MC part. So I'm going to change this annotation to MC. All right. So what do we do next with this? The real experimental process to do is that you should be cutting this plasmid with this enzyme and this enzyme. So let's just quickly cut it in silico using SP1 and BSPT1. If you hit run digest like this, it will show you that you actually will have two products from the digestion process. You have one that has like 2.1 kilobase and one that has around 160 base. You Benchling also generate this virtual digest for you so that they will have two bands in there, and you can actually compare whether it's going to be similar size or like different size compared to the other digestion if you have it in the same run. That was pretty useful because sometimes you have something that was closer to each other and you want to pick the right band when you are trying to do the downstream assembly. So I'm going to take this I'm going to take this sequence, copy it, and just do the DNA, let's just call it a texture of DNA from scratch, MC, RNA. I did Control-C copy already. This is just Control-V, and this is actually the backbone that we have got from the digestion of this PCK 411 right here. The insert, which is right here, has the part that we want to add into that sequence. I'm going to add right at the end of what we have right here. So with this, you see that the annotation of this piece and this piece were a little bit similar. The reason is because this piece is actually complementary to this piece to a certain extent. So if you search for this part, you see that these two are actually the same copy. So the way that the assembly overhang work is that they will show this part and this part, or this part and this part, depending on how each exonuclease were working. And the final sequence would be something that you combine these overlapping sequences together so that they turn into the original sequence. So if I take this combined sequence and I just do manual edit and go back to this plasmid and try to compare or search for it, it actually matched this sequence entirely. Again, we have to do that for the other side as well. So you can see that this is the extended part. If you search, you see this extended part up there as well. So what I'm going to do is I'm going to delete this whole thing. And you get the final DNA that was almost exactly as this plasmid by having just this 20 base that was changed using our new oligo, our new primer here that we ordered to make this new pizza fragment, combine and do the Gibson assembly or infusion assembly to make this new DNA piece. This will be useful for the targeting of the MC gene right away. All right, I think that's it. And the final thing I want to show you is the last kind of result that Benchling provide using the Golden Gate assembly strategy. So I show you that we have the gRNA cassette previously, right? a few minutes ago. If we try to make multiple gRNA cassettes using the Golden Gate reaction, the Golden Gate is actually like referring to the Golden Gate bridge, where you connect different parts together. So if you take a look right here, you can actually generate these parts and try to connect them together, similar to how the Golden Gate bridge were connected. One additional idea in the MoClone toolkit is that it's a lot easier if you introduce some selection process into these cloning or these assembly right away. So let's say that you start with the backbone plasmid that has GFP and you generate something that doesn't have GFP anymore. It will be a lot easier to select downstream because you can just pick the colony that was white. If there is any green colony still in your transformation, it means that that was just a backbone. There's nothing there that was new. So I think right now, if you were to clone 3 pieces of DNA into the plasmid of interest, I'm going to go into the Benchling and just show you briefly how can we generate this thing. But let's assume that we use other tools to design or generate these DNA fragment first. And the DNA fragment that I'm going to show you will have some compatible overhang right here, where they use the type 2S enzyme that is going to recognize at the red part right here, and then cut and give the sticky end that was specific, called LS, that can connect, that can attach to this one. The scenario is that there are 2 possibilities of connecting. This piece can actually come back and connect and make this, regular plasmid again if you do ligation. Or if this one come and connect, they will have no recognition site for the BSA1 or the type 2S enzyme anymore. In that case, they will just stay there and not get adjusted. And that's just like, one of the way of the Golden Gate reaction setup that would be useful at the end. If you're interested, please take a look at the Golden Gate protocol. I'll put like some link that was very useful to the recording as well. I think the site that I appreciate a lot, where I learned from, is the site from Jeffrey Barrick at UT Austin, like Jeffrey Barrick Lab at UT Austin, where they have like the wiki style that show how to do these processes step by step. All right, let's go to the Benchling part real quick. Let's see, Golden Gate. This should be number 4 instead. All right. So starting from this process, again, the Benchling is getting slower. If you open a lot of files, then it means that it's going to be processing that in the background. So most of the time, I'll just like close other tabs if I feel like the computer is running slower. With this, I'm gonna check the other 3 fragments of insert that I previously generated as well. So I have this 4', which is the backbone that has FFGP right here. I have 3 insert that has like spacer number 1, spacer number 2, and spacer number 3. These all look alike. They are almost the same. But the different part is these like 4 fragments right here that are different when they get cut by BSA1, which is type 2S enzyme that we use in this case. You see that like this type 2S part right here will be compatible with the type 2S part right here. And that's not easy for me to just like, go through it and like make sure that they're all correct. So I'm going to let Benchling help me with that. We're going to do Golden Gate. We're going to select the backbone. We'll use the full plasmid as a backbone and ask them to cut using BSA1. Now, the cut site on each side are AGZA or GACT on the bottom strand. We have 3 insert. I'm going to keep adding insert right here. The first insert, I'm just going to hit Control A, select everything, and ask them to do BSA1 cut site, set fragment. It said like, no, some of the backbone or insert are still unset. You have to keep moving forward. But what it showed me right here is that like this connection right here is actually correct already. So seems like it's good to go for now. Let's go for the second one. Let's add the second insert. All right, just do the same thing. It also give me a similar message that like this one can be connected properly. And if I do the last one, the last magical step, by setting this fragment. It just tells me that it looks like everything checks out. And you can just move forward. If you hit assembly right here and call them, let's say 3 gRNA assembly, it will give you this new DNA piece with some record right here that came from the backbone, which is the orange part, and the 3 insert, as shown by this color of like green, blue, and brown. I don't know what that color is. But yeah, so they also give you these assembly parameters on like, which design criteria were used, what is the basis that we use on the actual linkage, and its reverse complement. This is very important because the reverse complement can also be the same with the other bases right here as well. So let me show you one thing that will be bad for the assembly, so that like Benchling will show you that is not going to be correct. So I'm going to name them as TCGA. I'm going to change just these bases into something else. And then redo the assembly again. I'll go pretty quick. All right, backbone. We insert. First one is 4A. The second one is 4B. And the third one is the one that I just changed. So this one is going to be wrong. And look at this. Seems like they not let me assemble. They said like the dropout that this one starts with the second is GCA, but the final one ends with TCAG, which means this is not going to work. So whenever you do this design, you can also check whether in the in silico step, whether this cloning will work or will go smooth as you plan or not. And this is very useful because like sometimes we have like you're building up the inserts you want to put in, and it's going to be way harder and harder every time when you're trying to make something that was bigger. Sometimes like it's not going to give you an absolute no. It's just going to give you some suggestion like, hey, it seems like your sticky end are so similar to the point that like you may have some non-specific ligation, or like some products that you may not be desired, something like that. So that was a pretty useful situation as well. All right, finally, I think I wanna end by showing you that you can actually use the other function of Benchling apart from these like molecular tools as well, which is based on the notebook. So we have generated a few DNA pieces, right? If you make a note and you want to like link these DNA pieces into your notebook in Benchling, it is pretty easy to do so. So let me look for a fragment. Search for it. You can actually like link that one. You can make this kind of table by saying like, let's call it Ligate, let's say. And just try to link the fragment together. If this is the backbone, we use 4', 4E1. Then you find this one. If you hit that hyperlink, then it just show you this plasmid right away. Again, you can sort of like bring everything in. And you see, if your search term is too common, it's actually give you whatever available out there that you might find. 4C fragment. All right. So you can actually make this kind of report and link it. You can generate these like sharing link that you can share publicly with your friend. And that would be pretty easy or like convenient for us as a researcher or collaborative researcher, where we have to share this information all the time. And maybe finally, just want to show that you can, whenever you use Benchling for the real research process, if you want to get how do I say it? If you wanna take like new DNA from that gene, for example, let's go for maybe PCK 760 that we were talking about as a reporter. You can actually copy this link and ask Benchling to search for new DNA. And you can ask them to import from database and just paste the link right here. Once you search it, it will just give you that this is DNA that came from at gene. You can actually import it. And you get this DNA. It was similar to like what you're working on right away. Let's close this assembly for now. And another trick is that like sometime in this website or in AdGene, they actually like have some other sequence information that were provided. So this is from AdGene, right? But in this plasmid itself, they actually have one of the GenBank file from the depositor. So you can actually, just do the same thing, but upload the file instead. One key difference from these two processes is that if you take things from AdGene, the annotation would be based on what AdGene has annotated. You would take it from the depositor, it would be according to what the depositor has annotated. So, it depends. Most of the time, I appreciate the annotation that were given by the depositor much more than the auto annotation from AdGene. But the other annotation from AdGenes or like some other tools were pretty useful as well, because sometimes the maker of the plasmid doesn't really know some other part that they were using, and like some other thing actually show up when you do this auto annotation. So I think that's it for the basic of how to use Benchling, something like that. Hopefully I cover most of the basic things so far. If there is any question, please let me know. I can answer right now, or like I can just answer it like when the recording was up. If anything was commented on the recording, I can just like try to pop in as well. The three takeaways is that like, please make sure you use the molecular biology tools like Benchling or SnapGene or some other things to work on your DNA, RNA, and protein works. Benchling also has a protein function part as well. I didn't go through that because it's going to take a lot more time. But please use these kind of tools because like they, you should trust yourself as a scientist, but like having some additional verification would be much more useful when you work on it. The second one is like, please perform all in silico design and validation when you are doing this kind of cloning, because like, you may not notice this technical difficulty that may, occur down the line. Like, let's say when I do the digestion, if I just like copy paste without doing the actual digestion, I may not find some hidden digestion recognition site that were hidden in the backbone plasmid, and you ended up with 3 fragments instead of 1. So that could occur all the time. Let's say your plasmid has some mutations, if you don't know, and you just do digestion, you don't get the results as you expected. It would just be difficult in that step. And the last one is like, make sure you keep the good record of each DNA part. So having the table that linked the fragment that came from whatever starting plasmid together would be very useful when you do this kind of molecular biology works. Because again, if you just take a look at the DNA information, they all look almost identical because they're just ATGC, right? So you can't really interpret this information with just your eyes alone because it's so complicated. All right, I think that's it. I'll stop right here. And if there is any question, please feel free to jump in. All right, seems like there is no question. So thanks team for showing up and like, address any concern. I think I would just stop the recording now so that everyone can get back to work and maybe use some functions on Benchling to validate whether your design is going well or not. All right, let's stop the recording.